Good morning, grade 10 students. In this session, we're going to continue what we have started before about genetics polymorphism. We started first, we determined that the scientists proved that the nucleus is the carrier of the genetic information. All the information related to genetics, all DNA is found inside the nucleus. And to be more specific, it's found in the chromosomes and the nucleus. We said that the allele, it's a version or it's a form of a gene. For example, gene of eye color, we have different alleles. A gene of hair color, we have different alleles, and so on. We said that we have different types of cells. These types of cells, they are somatic cells and the sex cells or reproductive cells. These cells, somatic cells, they are all of the body cells except the gametes. However, the haploid cells, they are the sex cells and they are only the gametes, the sperm and the ovum. Where the sperm, it's the male gamete, it has the symbol, and the ovum, it has the female gametes. This is the female gamete, the ovum, and has the symbol. Beside the somatic cell, they have two copies of each chromosome. However, the haploid, it has one copy of each chromosome. It has two copies, so the number of chromosomes, it's 2n, and in a human, 2n equal to 46. However, in the sex cells or reproductive cells, the number of chromosomes, it's 1n, since we have only one copy of each chromosome. To continue with the types of cells, the different types of cells, we can move now to talk about this figure. These figures, they show haploid and diploid. Why haploid? Because we have only one copy of each chromosome, and the number of chromosomes, it's 1n. And always I repeat, 1n equal 23 in the case of haploid cells, the sex cells and the sperm cells and the ovum. In the case of the human, or in the case of gametes, sex cells, or it will be 2n equal to 46 in the case of diploid cell. Also, we are talking here about human. Let's move now to talk about diploid cells. We have two copies of each chromosome. They have the same gene, but they don't necessarily carry the same alleles. They are said to be homologous chromosomes. One of these chromosomes, as you said before, they are inherited from the mother, so they are called maternal chromosomes. However, other chromosomes, they are inherited from the father. They are called paternal chromosomes. So every chromosome, for example, pair number one, pair number two, pair number three, whatever, all of these chromosomes, one of these is going to be inherited from the mother, and one is going to be inherited from the father. The one inherited from the mother is called maternal chromosome. However, the one that's inherited from the father, it's called the paternal chromosome. Also, we said that the pair of homologous chromosomes, they carry the same gene but not necessarily the same allele. The two sister chromatids are said to be identical sister chromatids when they carry the same genes and the same alleles. So these, they are said to be identical sister chromatids when they carry the same gene and the same allele. Here we have homologous chromosomes. They have the same gene here, gene of the eye color. One of them is the color of the eyes is blue, and the other one is green. So they are a homologous. They carry the same gene, but they are not identical since they don't carry the same allele. We have green allele and blue allele. So in this application, we consider the gene of the eye color. If you have two alleles, green allele and the brown allele, what are the possible combination in different persons? So the possible combinations, they are brown and brown. So brown from the maternal and the brown from the paternal. So they are said to be identical homologous since they carry the same gene and the same allele. In person two, we have maternal is brown and the paternal is green. So they are homologous, not identical. In person three, the green allele is inherited from the mother and the brown is inherited from the father. What's the difference between these two? This is the difference that the brown here in person two is inherited from mother, it's maternal. However, in person three, it's paternal. And in person four, we have a green allele from the maternal and a green allele from the paternal. So they are said to be homologous, identical. So this person has two identical alleles. So whenever we have two identical alleles, the person is said to be homozygous, or we can say it's pure for the eye color. So whenever it carries two identical alleles, so the person is said to be homozygous or pure. Moving now, if it carries two different alleles, brown and green, for example, in this case, so it's said to be heterozygous or hybrid. Do you have to know both of them? Homozygous, it means pure, and heterozygous, it means hybrid? Yes. So whenever we have two identical alleles, I'm going to repeat, whenever we have two identical alleles, the person is said to be homozygous or pure. Whenever it carries two different alleles for the same gene, so this person is said to be heterozygous or hybrid. Person 3, we have green allele and we have brown allele. So this person is said to be heterozygous, or what do we call it by hybrid. So whenever we have two different alleles for the same gene, the person is heterozygous or hybrid. Whenever we have two identical alleles for the same gene, it's said to be homozygous or pure. This is the case of person 4, where it carries two identical alleles for the eye color. So this person is said to be homozygous or pure for the eye color gene. So here we describe what the meaning of homozygous and heterozygous. So when the alleles are identical, the individual is said to be homozygous, pure, or when the alleles they are different, the individual is said to be heterozygous or hybrid. Pay attention here. We are talking about the alleles of the same gene. Not, for example, you said we have the allele of the eye color, it's a brown, and the allele of the skin color is white. You said it's heterozygous. You can't work like this. It should be the same gene in order to say it's homozygous for the gene of the eye color. It's homozygous for the gene of the hair color, skin color, shape of the nose, shape of the ear, gene of the level of thinking, and so on. Now, let's talk about dominant alleles and recessive alleles. Always, we said that dominant is the one that dominates. Is the one that dominates, meaning that it is the strong allele. This strong allele, it can be expressed whenever we have two similar alleles, or one strong and one weak. That's why, because it's dominant, it is symbolized by a capital letter. So it's usually symbolized by a capital letter. And this is the dominant allele. The definition of the dominant allele, pay attention to this. Dominant allele is the allele that can be expressed in both homozygous and heterozygous. What does this mean? This means that it can be expressed whenever we have two identical alleles or two different alleles, since it's the dominant, is the strong, and it can be expressed in both cases. Recessive allele. Recessive, it's the weak allele. It will be expressed only in the case of homozygous. Why? It's weak. So it needs another weak allele in order to be expressed. That's why we gave it a small letter. So consider the gene of the eye color. If the brown, they said, allele is dominant over the green allele. Complete the following table. They said the brown is dominant. So the brown is the one that dominates. Who will be cited? So the one that is dominant, and the green allele is the recessive, is the one that is the weak allele. So here, brown allele, it's usually symbolized by capital letter since it's the dominant. So usually we can choose the first letter of the word. For example, in brown, the first letter is B. You said it's obligatory to do this? No. Simply you can choose N, you can choose W, you can choose Z. It's up to you. But in the question you have to follow the symbol. You don't have to change the symbol every one and then. Yeah, for example, whenever I took the symbol B capital for the brown and leaf, after 2 to 3 parts, I gave it symbol N. It doesn't work. You give it symbol B capital, you have to follow this during the whole question. So here we gave it the symbol B capital for the brown allele, and we gave G small for the green allele since it is the recessive. So here, moving to the table. Table, how many choices do we have? We have different choices. First, it can be BB, B capital, B capital. It can be BG or it can be BG. What is the difference between these two? BG, B is from the maternal origin first, and here G is from the paternal. In the second, the third case, or third person, here G is maternal origin, while B is paternal origin. And the last one is GG, it's the last case, G from paternal and G from maternal. So we have four different cases. Let's see, B is the dominant. So here we said that it can be expressed in both homozygote or heterozygotes. Where is the homozygote here? It's the BB. Where are the heterozygotes? It's the BG. So here B is the dominant, so the color will be brown. B is the dominant, so the color will be brown. And also here in the third person, B is the dominant, so the color will be brown. However, in the fourth person, it's a GG. And G small or green, it's recessive. So it should be expressed only in homozygous. It needs two weak alleles in order to be expressed. So this is the case here, and it's green. So this green allele, it cannot be expressed unless in the case of homozygote or pure. Instead, we have two identical alleles like this case. So here we have determined the appearance or the expressed eye color and the set of the eye color alleles. Here, the set of the alleles, whether from a maternal and paternal origin, we name it the genotype. However, the phenotype is the expressed trait. What can we see? So before we start with the exercise, I'm going to give the symbols. Here, symbols, let B capital for the brown. Since they said the brown dominates the green. So let B capital be the symbol of the dominant brown allele over the recessive green allele. And let G small be the symbol of the recessive green allele with respect to the brown dominant allele or with respect to the dominant brown allele. These are the cases wherever we have to give the symbols. In this part, I said, for example, designate by symbol the corresponding alleles. I'm sure I have to mention something in the text or some hint in order to know that this is the dominant and this one is the recessive. So let's continue with the table. In the table, as I said, we have BB, B from paternal and B from maternal. This person is said to be homozygous, or we can say it is pure, pure or homozygous. Do we have to know both? Yes. Here it's a pure, or we can say it's homozygous. Homozygous, okay? Pure or homozygous. And in the next one, it's BG. So it's a brown. Here in the case of hybrid or heterozygous. So you have to know both. Since maybe in the questions or the exams, I won't set, for example, heterozygous only. Maybe I'm going to set hybrid. So you have to know that heterozygous, it's hybrid, and the homozygous, it's pure. So both of these, they are BG, BG, they are heterozygous. Why did I put two? Because first one, P, it's from the paternal origin. This is the symbol of the male, you have to know it. This is the symbol of the male from now on. This is the male symbol. And you have to know that this is the female symbol and not the male symbol. So pay attention to this. Here, for example, whenever you give this symbol, for example, this is from paternal origin. And this one for maternal origin. By the way, these ideas already they are covered in grade 9, the same concepts, the same ideas. So here, paternal origin is from the father, they have to give it the symbol. And this is maternal origin from the mother, you have to give it the symbol. You have to pay attention to these ideas and these concepts. Already we covered them in grade 9 last year. but we are going to cover them in wide range in this year. So we talk about the paternal and maternal. This is homozygous. BG, it's heterozygous, and one from paternal origin and the other from maternal. So B here is from paternal, from the father, and here it's from maternal, from the mother. And the third person is the opposite. Here it's from the male, paternal, and this one is from the female, maternal. And the last one is GG, it's homozygous, and it could be expressed only in the case of homozygous. Let's continue to talk about the genotype and the phenotype. We said that the set of alleles is called the genotype, and the set of observed traits is called the phenotype. So what is the genotype? This is a Frayers model where here we give the definition. So this is the definition up inside the box. Here is an example. This is a non-example about the genotype. So here, what is the definition of the genotype? The genotype is the set of genes and their corresponding alleles. So the set of genes and their corresponding alleles is called the genotype. Example, BB, BG, GG, whatever. All of these alleles, the set of the alleles is said to be the genotype. However, tall, blue, small, large, all of them, they are not the genotype, they are observed trait. So here they are not example about genotype, they are example about the phenotype. Let's continue to go about phenotype right now. Phenotype is the set of observed traits, the result from the expression of the genotype. So the genotype will give the phenotype. Phenotype, tall, short, black, small, large, all of them, they are observed phenotypes. Blue eyes, red hair, for example, black hairs, all of them, they are phenotype. However, the non-example is BB, BG, GG. This is the genotype. So you have to know the definition of both the phenotype and the genotype. Now we're going to move to talk about the grafting. What is a graft? What is a graft? Graft is the transplant, either a tissue or an organ from one side to another within the same body or from one body to another. This is called the grafting or it's called transplantation. So grafting is the transplant of a tissue or an organ from one side to another in the same body or from one body to another. What do I mean by this? Let's see. Here, the graft is going to be done between two different individuals or within the same body. So what affects the fate of the graft? The fate of the graft is usually affected or depending on the similarity HLA. What is the HLA? It's the human leukocyte antigen. So here the grafting is affected, whether it's success or failure. Whenever we have a successful graft or a failed graft, so here it depends on what? On the similarity of HLA between the donor and the recipient. What the meaning of HLA? It's the human leukocyte antigen. This is human leukocyte antigen, human leukocyte antigen is found on the surface of the nucleated cells, human leukocyte antigen. So here, human leukocyte antigens, they affect the fate of the graft, whether it's accepted or rejected. So, what the meaning of the donor? The donor is the one that donates. Is here the animal or the individual that donates or that gives? So, for example, I'm going to donate a kidney, for example, for Ahmad. So, donating the kidney, I'm the donor, and Ahmad is going to be the recipient because Ahmad is going to receive the kidney from me. So here, this is called the donor and the recipient. What affects the fate of the graft? The fate of the graft is affected by the degree of similarity of HLA between the donor and the recipient. Moving now to talk about the different types of grafts. First type of graft is called the autograft. Auto, always I tell my students, auto is the first type. Auto, it's automatic. So definition, the graft is transferred from one side to the body to another side within the same body. What does this mean? This means, for example, I have to transplant, I have, for example, I burn my skin, let's say my face, I have a burned skin. So I have to transplant skin. So they get the skin from my thigh, okay, from my thigh to my face. This is an autograft. Why? Because the graft is done between different individuals belonging to the different regions within the same body. So here, autograft, the graft is transferred from one side to another within the same body. For example, from my hand to my thigh. Thigh, by the way, هي الفخذ. Okay? So here, from my hand to my thigh. However, autograph non-example from my hand to my friend hand. This is not an example, non-example about it. So here, first we have this is called the Frayers model, by the way. Already we talked about this before. This model is called the Frayers model. What does this mean? Frayers model, it works to give the definition and it moves to talk about the example and non-example. So to illustrate the differences between these two, it's called the Frayer's model. So Frayer's model, depending on this autograph definition, is the graph that is transferred from one side to another within the same body, and the example about this from my hand to my thigh, not example from my hand to my friend's hand. This is the first graph, and usually this graph is accepted since the cell within the same body they have identical HLA. Moving for the second type of graph. The second type of the graph is called the isograph. Iso, it starts with the letter I. So iso, it's done between two identical twins. This is called isograph. So isograph is transferred from one person to his identical twins. This identical twins is said to be identical, not fraternal twins. Fraternal twins, this means that this twin is going to be, for example, a brother and sister. And for example, this is called fraternal twins, and this is not an identical twins. So this is not the case, and it's not an isograph. So pay attention to this. The graph, whenever transferred from one person to the identical twin, is called isograph, and Samir's kidney transfer to his identical twin. This is an example about isograph, while non-example, Samir kidney transfer to his sister twin. So here it's not isograph, it's called fraternal twin. Fraternal twin is not like identical twin, and we don't call it an isograph. So here isograph always it's accepted. Why? Because the identical twins they have identical HLA. So the first two types of graph, the autograph and the isograph, they are accepted. These are accepted. So here they are accepted totally 100%. They have identical HLA. So allograft, the third type, is the graft that is transferred from one person to another in the same species. So transfer from one person to another. So different persons. So these two different persons, they are called allograft. Allo, different individuals, but they belong to the same species. What does it mean? Same species, human, for example, cats, dogs, monkeys. This is called a species. So from a human to human, from Sharif to Celia, for example, from Fatima to Haya, let's say it's an allograft. Why? Because they are different individuals belonging to the same species. All of them, they are human. While Another example from a cat to a dog. Cat and dog, they are different species, so they are not example about the allograph. Mostly, allograph is rejected unless you have certain circumstances and certain drugs they are taken, but mostly it's rejected because these individuals they carry different HLA. Moving now for the fourth type of graph. The fourth type of graph is called the xenograph. Xenograph is the graph that is transferred from one animal to another from different species. Different species mean that they belong to different species, like from a cat to a dog. This is an example. From a cat to a dog, they are different species. However, from Maya to Rula, it's an allograph because they belong to the same species. So here, as a summary about the graphs, we have four types of graphs. But before we talk about the four types, let's review together what is the grafting. So the grafting is the transplant of a tissue or an organ from one side to another within the same body or from one body to another. Here, the one that affects the fate of the graft is the degree of similarity between the donor and the recipient if they have identical HLA or if they have different HLA. The donor is the one that donates and the recipient is the one that receives. Autograft, the graft is transferred from one side to another in the same body, and the isograph is done between two identical twins. So these two identical twins surely they carry identical HLA. So these two, isograph and autograph and isograph, they are accepted. Why? Because the cells of the same body they have identical HLA. So autograph is accepted. Second, isograph, the graph is accepted since they have identical HLA for the identical twins. Allograph, the graph is transferred from one person to another in the same species. However, the fourth type is the xenograph, where it's done between different individuals belonging to different species. And this is everything for our session for